Hello. Today we're going to be going over neurons and the structures of the nervous system. This is a lab that I put together by Stephen Michelman. As an overview, the things that we'll be reviewing today, we'll have a background, history, a little bit for the nervous system, then our question hypothesis, our methods, our results, conclusions, the broader impacts, and then a page for our references or citations. As far as the background goes, well, there's many scientific truths that we simply accept as fact these days. Neuroscience had its humble, if not crude, beginnings, and much more recently than we generally assume. We just feel that science is always here and a part of our lives, but it hasn't always been around, and it hasn't always been as refined as it is now. As noted by Mo Constandi, for the majority of the 19th century, scientists could not agree on the basic nature of our nervous system. Some anticipated the neural networks with which we're familiar now, while others believed that the nervous system was just one large reticulum, which I always say is ridiculous. Let's see. Continuing in the background, as reported by Kendra Sherry, the number of neurons in the human brain is still a value frequently contested. And the usual, people for many years in the past have been saying 100 billion neurons. Lately they've been doing more research, and they haven't found an actual specimen of a human brain that has more than 86 billion. While people say, hey, well, that's just a 14 billion difference, it apparently makes, that is a significant difference. It makes more of a difference than people like to give it credit for. And the human nervous system is composed of many types of nerves and structures, and these are mainly divided belonging to the peripheral or the central nervous system. For our question and our hypothesis, we're asking how many identifiable structures constitute our nervous system. I believe that all the currently identifiable structures of importance involving our nervous system can be isolated, identified, and photographed. And that's basically what our lab activity is over today. This is the website to which you can go or link. And by going there, you can observe the images of all the relevant structures in the nervous system, those that comprise the central and peripheral nervous systems anyway. These images are captured by slicing and separating the brain into various constituent parts. Then they're stained with various compounds to make the smaller structures within them visible. They provide contrast with these stains. Images are captured at anywhere between 4 times to 10 times 100 times magnification. And lenses like the 100x lens would also require an oil immersion. This allows it to achieve a greater degree of resolution for its magnification. There's a lot of little nuances in the actual imaging that go into it. So the different types of stains that we used in this lab, or that were used in this lab to bring about these images. We have a silver stain, known as the Golgi stain. It's the oldest and most sensitive stain in neuroscience. A small subset of neurons are completely stained, allowing visualization of complex dendritic trees and an exploration of the immense diversity in neural shapes. So that's a very good stain. The fun fact is, while Golgi himself was a proponent of the reticular theory, thinking that the nervous system was just one large structure, Santiago Ramon y Cajal used Golgi stain to provide convincing evidence in support that the neuron doctrine by which we live today. The hematoxylin and eosin stain is a histological mainstay. Sorry, some of these hematoxylin, I very rarely have to say those words. Your pathologists go to. The H stain stains nuclei blue, while the E stains many proteins red. The staining allows for easy differentiation of tissue types. Continuing with the stains, we have the azan stain. It uses azachromine G and alkaline counterstain. It dyes the nuclei and acidophilic structures red, while basophilic structures take up the counterstain, frequently aniline blue or orange G, especially suited for visualizing connective and epithelial tissues. We have the osmium tetroxide stain. It binds lipids and is frequently used in transmission electron microscopy. In which application it inserts heavy metal into lipid structures such as cell membranes, enhancing contrast. It gives a dark color and it's great for viewing myelin, like myelin sheets. Everyone loves myelin. I mean, if you like neurons, you like myelin. The Nissl stain binds RNA, allowing visualization of nuclei and cell bodies where rough endoplasmic reticulum and ribosomes are present. It's a principal stain relied upon by neuroanatomists to match experimental tissue with brain atlas. So, if we were doing this research, we would need to know all those stains, but we would also need to know what our microscope is and all of its constituent parts. So we have an image here of all the different labels, and hopefully that would help us use that. But since all of these images were done for us, we don't really have to utilize it today. As a result, of the imaging, let's see, we can definitely identify all the structures of which we're currently aware existing within the peripheral and central nervous system. I have a list here on the side for the peripheral nervous system where we're able to find the motor endings, receptors, autonomic ganglion, dorsal root ganglion, and peripheral nerves. We have a couple of pictures here of the motor endings and the peripheral nerves. For the central nervous system, we were able to isolate the cerebrum, cerebellum, spinal cord, neuroglia, and medulla. And we have a couple of images here. We're able to see all these structures from various angles and various magnification intensities. In conclusion, our hypothesis, though limited in scope, was correct. Using the virtual lab as a resource, we're able to see all the mapped parts of the peripheral and of the central nervous system. The next step might be to use the antibody stains to see what chemicals, like dopamine or acetylcholine, are in what structures. Broader impacts. There's broader impacts to be considered. As noted by Leo Winnerman, the discovery of mirror neurons was considered by some the most important story of the decade. I mean, this is mostly by psychologists and psychiatrists because, the mirror neurons were great at explaining some things that we hadn't understood before, and it just opened up another page of research and things to look into. Understanding the brain circuitry underlying addictive behaviors has led to the recognition of addiction as a medical issue rather than a moral failing. Functional anatomical imaging has allowed Adrian Owen's group to communicate with people trapped in vegetative states. Further understanding of our neurology will also have implications for those suffering from Alzheimer's, sciatica, chronic pain, epilepsy, and many other various nerve and neurological conditions. Don't you find it inherently valuable to understand the little pieces of the clock whose ticking is your subjective experience? Then we have some citations and references, and that pretty much concludes my presentation today. Thank you very much. Sorry for going over time.